I'm going to introduce our panelists in the order in which they were put on the website, not necessarily the order in which they are presenting. So I'm going to start with Rika Anderson. Rika is an assistant professor in the biology department at Carleton College with a great amount of deep sea sampling experience as part of her ongoing research project surrounding microbial ecology and evolution in deep sea hydrothermal vents. Much of this research has been conducted in collaboration with her collaborator, Julie Huber, our next panelist. Julie is a senior scientist at Woods Hole Oceanographic Institution with vast experience in deep sea sampling at hydrothermal vents and in oceanic crustal fluids, like those found in the basement at North Pond near the Mid-Atlantic Ridge. Her research focuses on how reactions between the ocean and the seafloor create energy to support microbial life in the deep, dark ocean. This energy is largely created abiotically, which relies on geochemical cycling. This segues nicely into our introduction for our next panelist, Susan Lang. Susan is an associate professor in the School of the Earth, Ocean, and Environment Department at the University of South Carolina. Her research into water-rock microbe interactions and aqueous biogeochemistry allows a deeper understanding of how life finds a way in sites like those in the Atlantis Massif subsurface and in the Lost City hydrothermal vent field. Our final panelist is Michael Rappé. Michael is a research professor at the Hawaii Institute of Marine Biology. His lab uses diverse approaches, including molecular biology, genomics, and traditional microbiology, to understand the processes that facilitate microbial life in basalt-hosted deep subsurface crustal fluids in places like the Juan de Fuca ridge flank in the Northeast Pacific Ocean. Like all of our panelists, he has a lot of experience in using pumps and samplers and different pieces of equipment that are able to provide a window into the microbiology and the geochemistry in the seafloor. Collectively, our panelists bring a great amount of knowledge to the table regarding sampling both fluids and sediments at hydrothermal sites and the cold crustal biosphere. So today we're actually going to be starting with Julie.
Great, thanks, Joy. I've been given the job of providing some overview for all of the speakers, and then I will get into the samplers that I'm talking about. And feel free to ask questions in the chat, even if it's like, what ID in tubing do you use? Because that's where we're at when we really start using these samplers. So why do we sample crustal fluids? Well, for a lot of reasons. But we know that seawater moves into oceanic crust. It often gets modified through the crustal aquifer. It comes back out somewhere else. But we rarely know where recharge is happening, but we often know where discharge is happening. So by sampling these discharging fluids, we can learn a lot about what's happening within these reaction pathways beneath the seafloor. And so these crustal fluids are windows into these subsea floor processes. Now, there's many flavors of crustal fluids, depending on the tectonic setting or where you are in the ocean. Some of them are illustrated fluids, as well as a lot of different types of hydrothermal settings, and probably even more that we haven't yet discovered. And, a lot of us are oceanographers. And so we learn that there are a lot of different ways to sample water in the ocean, right? But today we're really going to be focusing on seafloor sampling of fluids that are coming out of the crust or the sediments in the case of some of our ridge flank work. There's a whole myriad of tools that we aren't talking about, just so you know. This is a table that Susan Lang put together of the samplers, some of the major samplers that we're going to be covering today. Not just major samplers, large samplers, I guess is the word I should be using. And sort of, the volume of fluid you can collect, how many samples you can get per dive, what the method is for the collection, whether or not the samples are gas type, whether or not you can do in situ preservation, and who to contact. So one of the reasons I did want us to record this is so that you could go back and look at this table if there's someone you want to follow up with about this. So I'm going to start sort of in the middle with this super sampler and talk about one other sampler. I'm going to have Rika jump in and talk about the UFO. Then I'm going to hand off to Mike, who is going to talk about the mobile pumping system. and then over to Susan to talk about the hog and some of these community assets at the top. So my disclaimer is that I have built none of these things I'm talking about. I am a tried and true microbiologist, but my entire career, I have collaborated with engineers and scientists and basically modified and used their samplers for my research. For me, this has been incredibly productive and has just like created a lot of new avenues for research and innovation by working with these folks. A few years ago, I finally did buy a sampler and I still haven't finished putting it together. So I feel like I have a lot of experience in the sense of not only using these things at sea, but also in collaborating with people to figure out how to get them to work for the science you want to do. So the first sampler I wanted to talk about is the super sampler, which is the suspended underwater particulate rosette sampler. This was created by Chip Breyer. He was at Hui at the time. He is now at UT Rio Grande Valley. He's both a geochemist and an engineer. And this was originally designed with plume sampling in mind. So he was really interested in filtering large volumes of water from the buoyant plume of a deep sea hydrothermal vent to look at mineral precipitation, geochemistry, and with colleagues, microbiology. And the idea was to mount this instrument on an ROV and move up in 2-meter increments through a hydrothermal plume. It's though one of the most versatile instruments out there in the sense that it's been deployed on an ROV, an AUV, and even a hybrid vehicle. The ROV version has a 14-port valve, which is what is shown here. And then it's paired with a McLean pump. And it's really the valve that is distinct about this instrument. You'll hear the word McLean pump a lot. McLean is a company based here actually in Falmouth that makes a lot of the pumps that we use in oceanography. And it's really flexible in the sense of whether or not your end effector is a bag or a bottle or a filter. And this instrument, you can now purchase it from McLean, meaning you can purchase the valve and the pump and some of the instrument electronics. And this is just a picture of a variety of the configurations I've been to see with Super. This was some of the first tests. This is the front basket of the Jason remotely operated vehicle when they were testing a bunch of different things for that plume sampling. The image I showed back here is actually from the world's deepest known hydrothermal vents at 5,000 meters. Over here is when we were revisiting some of those sites and we were using the Nereus ROV, but also an autonomous underwater vehicle, and he mounted the sampler up here. He's also put a version of this on the Sentry AUV, and then most recently, my team used it on the Ocean Exploration Trust ROV Hercules. So again, this is a really flexible platform that can go in a lot of different spaces, which is one of the reasons I wanted to include it. Another sampler that Chip developed that I helped him test at sea is this precision multi-sampler. We just call it the MAT sampler, but I included it because when we tested it, we used it for fluids as well. And this is a really clever system. You know, as you'll see with a lot of these, a lot of them have pump, electronics, and then it's really these end effectors that vary. And it has these magnetic swappable magazines, which are pretty cool and fun to use, unless there's a lot of iron around where you're working with, and sometimes the iron gets all over the magnets. But ultimately how we used it is we pumped water through these Steravex filters, or we just sucked up water into these chambers, or we sucked up bits of microbial mat. And this is an example of us sampling these thick iron microbial mats at the Mid-Cayman Rise. So this is also published in Deep Sea Research if you want to learn more about it. And here you can see the iron sticking to the magnetic magazines that come out in these 6 holders. So the next thing I want to do is hand this off to Rika to talk about the UFO sampler. She has 2 slides in here. Rika?
Yeah, thanks, Julie. Okay, so I'm going to be telling you all about the Universal Fluid Obtainer, or the UFO. And to give you some background on this sampler, this came out of conversations that a bunch of early career folks were having about the fact that many of us who were starting off our careers didn't really have access to many of the fluid samplers you're hearing about today. And we were, invited on cruises and we needed fluid from the deep sea, but we didn't know where we could get a sampler, or maybe we don't know Julie or don't know Susan, and we couldn't just call up and say, can we borrow your sampler? So we realized there's this need for a community resource that anybody can tap into if you are using, say, an ROV or an HOV to sample from the deep sea. And so that's what this came from. So we worked with the Jason and the Alvin groups to put together a sampler that you can see here. It was designed by Andy Billings, who's one of the Jason techs. And he put together a valve that you can see here that has 8 sample ports. One of them needs to be used for flushing. So that means there are 7 ports available for sampling. You can see a top view and a side view. It's meant to be really flexible. And so you could hook up, say, a Stervex filter, like Julie mentioned, to it. You could hook up bags to it. And it's meant to be kind of like a plug and play sort of system. So this is probably the simplest sampler that you will see presented today, but it's also meant to be one of the most adaptable. So you can see pictures here from its maiden voyage out on the Mid-Cayman Rise last 2 years ago now, where we had put both Stervex filters and a bag onto it. We literally stuck the bag in a milk crate next to the valve. It's not very fancy, but it does the job. And so if you, let's see. Yeah, so if you go to the next slide, I have a few more details. So it's meant to be a flexible and adaptable according to the user's needs. So Andy says, think of it as like a kit of parts that you can use to mix and match. Right now it has lure lock fittings on it. And so we did that so it could be really flexible, but that actually can be changed as well. And like I said, you can fit it with Cakeabads, Stervex filters, all kinds of things. It does need a sampling intake. So when we were out 2 years ago, we used Susan's probe that we use to actually take the fluid in, but we're working on that right now. Currently, it's a positive pumping system. So we pump fluid into the filter and into the bags, although we've been talking about changing it up with more of a displacement system, which I think somebody else will be talking about later on. The most relevant information for you folks to know is that if you want to use the UFO to do sampling of the deep sea, you can include it in your proposals to NSF, as well as in your ship time requests for use with either ROV Jason or HOV Alvin. And I would recommend you also get in contact with the deep sea submergence folks who are responsible for those. So Matt Hanks for the Jason group and Bruce Strickrat for the Alvin group, just to let them know that you're interested in using the UFO for an upcoming cruise. All right, that's all I had.
Thanks, Rika. So the other thing I wanted to mention is that a lot of these systems allow for in situ preservation of filtered biomass. And as we were in our breakout group, I realized I have some of these in my office since I'm getting ready to go to sea. So this is one example of how you can filter through microbial biomass. It gets caught on the filter, and then you can add a fixative of some sort. In this case, we usually use RNA later, but there's a lot of different sampling gear out there. The one in the middle, these are actually 142 millimeter filters that Chip Breyer and Greg Dick developed for their plume work. And there's a couple other ones out there. So again, if that's something that you're interested in, there are, for example, I have lent out my filter holders to a lot of different people over the years rather than have them buy their own. So I just wanted to close with a few slides about some not as complicated and fancy tools, but that some of them are really standard on a lot of vehicles that we use. So one is the vacuum cleaner of the deep sea, which we call a slurp or a suction sampler. Most vehicles come with some version of this. And this is a drawing from Cellular Robotics, but this is actually the slurp sampler that's on the ROV Sebastian that Schmidt Ocean Institute runs. And it literally is usually a vacuum hose like this. It has a big fast pump, and it was really designed for collecting animals in microbial mat, but I've been at sea before when our main fluid sampler has broken. And in an area where you have active flow, it's actually worked pretty well for getting a bunch of water to keep things going. And so it's just something to think about in terms of asking a provider, do you have a slurp sampler? It's always good if they do, even if you're not thinking you're going to use it, to maybe bring it out in case it could be useful. Also, another tool that a lot of vehicles have are pelagic pumps. And again, there have been cruises like Rika where I didn't have access to a fluid sampler, so I just sort of jerry-rigged something out there where I could move a lot of fluid through a pump. This is a Stera-Pak filter that goes through this pump. One of the disadvantages is that your sample does have to go through the pump. Otherwise, there's just too much resistance. But again, if you wanted to move through the water column and filter 1,000 liters of water while the ROV is doing other things, this is a great way to do it. And then finally, this is a squeezer syringe sampler that was developed by Jeff Wheat, and both he and Beth have used it and provided me with these images. And this is a way to get small volumes of water. It's using the manipulator of the ROV or the HOV to trigger it. And I think you get about it. It's like these giant veterinarian horse syringes that I think are about 150 mils. But again, for some basic cell counts and chemistry has been provided really, really good samples. So with that, I'm going to hand it off to Mike to talk about his MPS sampler.
Okay, just making sure everybody could see that title slide.
Yes.
Okay, so yeah, so I'm going to talk a little bit about the mobile pumping system, or MPS sampler, that we use to collect fluids using positive connections to corks, or circulation obviation retrofit kits on the seafloor. Sorry, Jeff, but there you are. One thing I'm going to mention right off the bat is the same similar disclaimer to what Julie mentioned, in that I'm a microbiologist, and while I enjoy using fancy tools, I have not been a part of designing the mobile pumping system. This was really the baby of Jim Cowan and Brian Glazer as a postdoc with Jim, and Jeff was involved, and also Tina and Oliver, who were intimately involved in designing the mobile pumping system and a large variety of associated and periphery equipment as well. Anything you actually want to know about the mobile pumping system, this publication does an excellent job of laying it out, including mechanical drawings. Really, anything you want to know about the mobile pumping system is found in this publication. So it's an excellent resource, not only for the mobile pumping system, but also a lot of associated kind of downstream from the mobile pumping system samplers, as well as some more complicated platforms that we maintain at the University of Hawaii. So really, in a nutshell, the mobile, come back. Like a lot of these samplers, the mobile pumping system, it's pretty straightforward, right? You have your electronics, you have your mobile pumping system controller that interfaces with your ROV or AOV. You have a pump, the mobile pumping system pump, which I'll talk about in a little bit more detail. And then we include consistently an oxygen optote, a flow sensor, and a temperature sensor. And this is so we can monitor in real time, basically, how our connection is doing to the cork, which is another important aspect of this that I'll just talk about in a little bit more detail. So we connect to the cork through an umbilical, and connectors designed specifically to give us positive connections to the cork to exclude any bottom seawater. Because one aspect of this is, we're sampling fluids that have very low biomass. I mean, we can, many of us also work in the water column, and we consider the deep sea low biomass. Well, the cork fluids are often an order of magnitude less biomass than that bottom seawater. So any seawater intrusion into your sampling can just wreak havoc on any downstream, for example, molecular analyses. The mobile pumping system acts as kind of a front end to really, whatever you can imagine for a collection mechanism for the fluids, whether that be filters, whether that be bag samplers. And I'll just mention a couple of those kind of downstream collection ways we've used to collect the fluids as well. So this is a shot of the mobile pumping system. The mobile pumping system is here on the left-hand side, actually. And you can see right center, the umbilical and the connector that we use to connect to the port, to the cork. And then on the right-hand side, we have just one variety of our bag samplers that we use consistently. And this is a large volume bag sampler that can collect up to 70 liters of crustal fluid right there. So I'll go into a little bit more detail about what you're, but I just wanted you to see this on the front basket of Jason. Okay, so as I mentioned, we have this umbilical that we use made of Superflex tubing. And we have what I refer to here as the Hans connector. This is referred to by many names, but this is a connector that we use to connect directly to the cork. And I think, yeah, there we go. And also, as I go through this, really quick disclaimer, you may see one of your own photos in here, you may see one of your own images in here, and I very much apologize that most of this is not credited to their source. I've taken a bunch of this from a gazillion other PowerPoint presentations designed by other people, and so a lot of these aren't given credit, so I just wanted to mention that. This one actually is. This is from IODP proceedings for 327. So shown here on the left is a bay of ports on a cork that access different fluid delivery lines from different horizons in the deep subsurface basement. So these have been designed to match with some of the connect. Oh, God, yeah. Sorry. Some of the connectors that you see on the right-hand side here that are connected to our mobile pumping system, so we can make these positive connections to the cork and to the fluid delivery lines. Because like I said, seawater is always the bane of our existence in this case. So just a different shot of the mobile pumping system here. We have the controller kind of in the center there. That's our electronic interface. You can see the what we call the mega pump. It's a custom designed pump that I'll describe for you. And the seabird temperature sensor. Buried in here a little bit is the flow sensor and the oxygen sensor. I don't really talk about it much, but on the left-hand side of the mobile pumping system, you see like another cylinder. This is actually a fluid trap so that we can use gas-tight samplers to actually sample from an adapter that fits inside that cylinder there. It's connected to the mobile pumping system. I kind of consider it a little bit separate because usually it just has this cap on it and that's it. But if we specifically want to sample using airtights or something similar, we have an adapter that fits in there. So the mega pump is not very photogenic. It just looks like a gray cylinder. And so this is from the final mechanical drawing for the mega pump. The thing about the mega pump, we had to design there's just nothing off the shelf that would function to pull the volume of fluid. The mega pump here pull 5 liters of fluid potentially through a fluid delivery line at the seafloor. There's just nothing off the shelf that fit all the requirements for this pump. It's a positive displacement pump head, and that we had custom made with PTFE and titanium wedded parts, so inert parts, that's been magnetically joined to a brushless motor. We had a custom designed oil filled housing and motor. And then the magnetic coupling really eliminates the need for any sort of shaft seal, which was also important. So this is described very well in that publication I mentioned. So if you're interested in designing anything similar, I really urge you to look at the publication there. So once we collect those fluids, really, like a lot of, what several things Julie mentioned also is that what you do with the fluids downstream is really kind of like up to your imagination, right? For some things we do, we like to collect large volumes of crustal fluids, so that we can do a lot of analyses in parallel in terms of biogeochemistry, in terms of rates, in terms of molecular biology. So we have samplers, like I mentioned earlier, that at the top here is a large volume bag sampler. So each bag, we can deploy 2 of these at once, and each of them will collect 70 liters of fluid for you. On the bottom left, we also frequently will hook up to a McLean sampler with 24 ports on it, and we can use that to direct fluid into several different parallel bags in what we call the medium volume bag sampler. So each of the bags on the bottom left-hand side, and this is mounted on the rear of, what is this mounted on the rear of? This must be Jason also, I think. This medium volume bag sampler has 6 different bags, and you can see these are actually foil bags. And then we can also adapt the McLean sampler. So the bottom right-hand side, you can see a big mess of Sterivex filters there. So we can also direct the fluids through Sterivex filters, and all this is controlled in real time, can be controlled in real time, so that you can move from port to port, decide how much fluid you want to put through any individual filter or any individual bag. And then the top right hand side, we have another variety of bag sampler. These hold 500 mil. This is kind of adapted from an off-the-shelf thing from McLean, but these hold 500 mil bags. And we frequently will use foil bags in here also, so for gas analyses. So one thing I wanted to mention is we frequently use the mobile pumping system attached to an ROV or a AOV and control that in real time, which is great because then you can monitor the temperature, you can monitor the flow. But then, the mobile pumping system is adaptable enough to where, you can also, with a different controller, program it, deploy it for autonomous sampling as well via elevator. And that's what I just threw this in here to remind myself of that you can see kind of the fluid delivery line. You can sort of see it, that blue squiggly line connecting the elevator that contains the mobile pumping system to the cork. And I think that's it. Yeah, okay.
Okay, I will try and share my screen as well. Let's see. Can people see that okay now? Great. So I'm going to talk a little bit about not just the sampler that I use, but also the ones that are not represented by people here. One of the very earliest samplers used to collect hydrothermal fluids is called the major samplers. And so you'll hear a lot of us refer to major samplers quite frequently. This was first used, they were really designed to be used to collect very hot temperature hydrothermal fluids. So they're made entirely out of titanium, and they're basically large titanium syringes with an O-ring. And so there's a diagram down here, and this is a picture of 2 of them being deployed in parallel. And so this is the syringe before it gets triggered, and then the syringe after this spring is triggered. And it just withdraws a piston and collects up to about 750 mils of fluid. Since it's entirely titanium, you can use it in temperatures up to 400, 450 degrees. And so you can use it in pretty much any hydrothermal fluid. But since it's entirely titanium, it's also been found to be really good for things like trace metals and stuff like that. The drawbacks compared to some of the other samplers, particularly for microbiologists, are that the volume is relatively limited, and it's not gas tight, by which I mean if you have something with high concentrations of hydrogen or methane, those will separate into a separate gas phase, and then you'll have a tough time quantifying them. And so let me just also mention that these, if you are interested in using these, there are several owned by both the Jason and the Alvin groups. So again, anyone can request these when you do your ship time request or when you write your proposal, and just say you want to use major samplers, and then the groups will provide them. You'll be responsible for prepping them, cleaning them, and turning them over. And prepping them can be a little tricky, but again, people know how to do it. In this case, there is a temperature probe that is put in conjunction with where the snorkel is. This is the snorkel. This is obviously where the fluid is. And then it's being triggered right here by the arm of the ROV Jason. And this here is helping to read the temperature. It's an ICL link, and that is allowing feedback in real time on the temperature of sampling. And that temperature feedback in real time is really important in systems like this, because there can be centimeters of difference between where the fluid flow and where you're in background seawater. And so, as Mike has mentioned, and other people, trying to minimize the entrainment of seawater is really important. Another kind of early on sampling device is called a gas tight sampler. And this works by creating a vacuum. This is also basically a titanium syringe. You create a vacuum within the chamber. This is your snorkel. And it's also triggered by an actuator valve. There's no temperature probe that's incorporated into this one. And then the samples are extracted at sea, usually at sea. Sometimes if there's no extraction line at sea, you can ship the samplers back and do that shore-based. You just can't reuse the samplers again in that case. But and then the gas is collected and stored in glass vials typically. The benefit of this is that it keeps your samples under pressure, and so you can quantify volatiles that would degas otherwise. So as your sample is going from the high pressure and high temperatures of its kind of in situ conditions into your shipboard lower atmospheric pressure conditions, then you'll start getting bubbling and degassing, and then it's impossible to quantify what the actual concentration of your gas is. And so these are pretty much almost entirely with Marv Lilly at UW right now. In 2018, this is Tamara who's on the line, a larger volume of these. So the typical ones are 150 mils, a Titan version to collect very large ones so that the clumped isotopes of enough fluid for clumped isotopes of methane could be collected was designed and built. And so there's a couple people out there who know how to use these and can borrow them and use them at sea. But Marv Lilly is kind of the central go-to, although probably Tamara would also be able to answer more questions about these than me, for sure. Another version of a sampler that can keep gases under pressure and be used to quantify actual concentrations and isotopes of volatiles that would degas otherwise was designed and built by Jeff Seewald at Hui. And so these are called isobaric gas tights, and a little diagram of how they work is here. Instead of having them under vacuum, you use compressed air to keep the sample under pressure as it's being brought back up to shipboard conditions. These are also designed to be sampling fluids up to 400 degrees. Everything's titanium. One of the benefits of them is that you do not need a extraction line at sea. And so when they come back on deck, you sample from them using by repressurizing them in a reverse direction using an HPLC pump. And they have a temperature probe incorporated into the snorkel intake. Jeff Seewald at Hui has these, as does, I think, Jill McDermott at Lehigh University has some of her own, and Owen Reeves in Norway has some, and there's probably a couple other people who have built some. So if you really want good quality gas samples, you really have to go with one of these two, unless you're looking at least if you're looking at fluids with high concentrations of gases. If you're looking at fluids that have gas concentrations that are not super saturated at room temperature conditions, then you don't have to worry nearly so much about this stuff. But for high concentration gas stuff, you really have to go with either an IGT or a gas type. When I was in grad school, I used, Julie mentioned the hydrothermal fluid and particulate sampler, affectionately known as BEAST, which is the circa late 90s, early aughts version here. And it used to be deployed kind of in what we would call the front basket of the vehicle. And here's the intake here, and then the sample chambers are here. It's undergone some major redesigns over time. And this operates with we've kind of talked about the difference between positive pumping or displacement, but haven't really explained what that is. So the displacement method is that you take these chambers and you put a bag inside of them and then fill the chamber with water. And so they go down with these chambers filled with water in an empty bag. There's fluid that goes through this titanium stem that's actively pumped through this titanium stem. And then when you want to sample, what you do is you actually just pump the water out of the bag. And that causes, because of the displacement, it causes water to be drawn. You pump water out of the chamber, and that causes your sample to be pulled into the bag. And the reason for doing it this way is that then your sample is only ever exposed to the intake line where you can control the materials or the fittings and into the bag. And so Mike mentioned that, the pump that they built was custom built to use materials that would not contaminate for things like organics and other things. And so that's one approach to making sure that the materials that your samples are exposed to are really, really clean. And then this is a different approach. So in this approach, you don't need the pump to be really clean or anything special. But the downside of the displacement approach is that you define the volume that you end up with by the volume of the chamber. And so by positive pumping, the benefit is you can throw that bag into a milk carton and fill it up, and it doesn't really matter. In this case, if you want a 750 ml sample, you have to design a leak tight 750 ml chamber that you can pump water out of. So hopefully that makes sense. And this is a schematic of the sampler that I built that was largely based on Dave Butterfield's fluid sampler. And so the schematic is very similar to what his would look like. And so I figured I'd just go ahead and use that instead of trying to find one of his. But on mine, there's two different inlets. The fluid pathways for biological samples and for chemical samples is entirely separate, because I go to see with microbiologists that want to fill these chambers with preservatives. And even though we have check valves that should prevent sample from escaping the bags and going into the fluid pathway, they're sometimes imperfect, and if that gets into the chemistry lines or the chemistry samples, then you can get some pretty bad cross-contamination. And so the way it works is that you have sample, your intake is put wherever you want, and then you positively pump through these major titanium stems using a 4 liter per minute pump. And then when you're ready to take a sample, you would turn your valve to whichever chamber you want to sample, and you're really just pumping water out of that chamber, which is drawing fluid in from that titanium stem. And so whatever you connect to that titanium stem is what you get in terms of sample. And so we built some large volume boxes that sample 9 liters. Our regular chemistry and microbiology samples are 2 liters, and then you can connect filters, and for those you can basically filter until you either get bored or the sample, the filter itself clogs. And we've done Sterivex, and we've borrowed some of Julie's RNA preservative filters as well. And so this is just what it looks like. This is kind of the main body of where the chambers are and where the fluids would be. This part sits on the back of the vehicle. We've only deployed with Jason so far. It can be reconfigured to deploy with Alvin, but we haven't had to yet. And so but I'm sure we can. And with the intakes, this is the intake. Again, we have 2 intakes, one with biology and one with chemistry. And there's a temperature probe that's incorporated into the intake so that we know exactly what type of fluid we're collecting, and we can sit there for an absurd number of hours and minutes saying, can you put can you move it a little to the right, a little to the right, a little to the right. And then, like Dave's samplers, these can be, the actual chambers themselves come off as trays and can be subsampled as trays. And so these are some of Billy Brazelton's group kind of working with the biology samples, and you can filter straight out without ever exposing your sample to air. And then Sarah Hugh and some of Julie Huber's group, and then other microbiologists have used the microbiology chambers and filters to subsample. And then this is a slide that Julie provided, and is showing some of the work that's been going on with Dave Butterfield's sampler to modify it so that you can do some in situ incubation experiments and some experiments on the seafloor. And so you can, with some of these modifications, you can keep the sample chambers at temperature and allow different experiments to be carried out. And Julie, if you have more to say on that, I can let you finish up on that.
Is this your last slide, Susan?
This is the last slide, so.
Okay, yeah, I just thought it was fun to put this in at the end, just to remind people that like all of these things being described are super flexible in the sense of like, what is your idea and what do you want to do? And you know, this was one example where after 15 years of using this sampler, we decided to try to do this experiment on the seafloor. Another example is those IGTs that Susan showed from Jeff Sebald's lab. He worked with Stefan Sievert's group, and they collected the fluid, they brought it back on deck, kept it at pressure, added tracers, and looked at microbial carbon fixation at in situ pressure. So I wanted to put it out there, as there's a huge amount of literature also on doing these types of experiments, modifying these samplers.